Hello, welcome to our video that demonstrates the procedures for collecting water samples for the invasive carp eDNA sampling methodology in accordance with the U.S. Fish and Wildlife Service Region 3 Invasive Carp eDNA Program's Quality Assurance Project Plan, or QAPP. For the most comprehensive information on how to sample eDNA, please refer to Section B2 of the QAPP. A sample plan should be made prior to sampling. It is important to ensure all crew members are aware of what their role will be and what is expected of them for each study site. At a minimum, a crew should consist of a lead sampler, a sampling assistant, and a boat driver, viewed from left to right in this example. All crew members are responsible for reading and familiarizing themselves with the QAPP prior to departure. It can also be beneficial to have the sample coordinates entered into the boat driver's GPS prior to sampling, but it is not required. At the very least, detailed maps should be included in the sample plan. Prior to sampling, crews will need to pre-label all the tubes that will be required for sampling. The tubes will be placed in clean coolers in a fashion that allows samples to be collected sequentially. Depending on the sampling design implemented by each case number, the number of samples in a cooler and the number of coolers may vary. Additionally, prior to launching each day, the hull of the boat should be sprayed with a bleach or Virkon solution and allowed to sit for the appropriate contact time. It is also important to fully decontaminate all gear and equipment between sampling of different cases to mitigate for cross-contamination. The lead sampler begins by mixing a 20% bleach solution. While doing this, the designated data recorder, who in this example is also the sampling assistant, makes sure their iPad is up and running, the field map app is functioning properly, and the GPS is accurate. The boat driver maneuvers them to their appropriate starting point, either the downstream-most sample point in a Lotic system or downwind-most sample point in a Lintic system, to ensure the surface isn't disturbed by the boat during sample collection. In front of the sampling assistant is the clean cooler that contains the pre-labeled tubes, while in front of the lead sampler is the quote contaminated cooler, where the collected samples are stored on ice until sampling is completed. To the side of the lead sampler is the container of 20% bleach solution to disinfect sampling racks between samples. Before sampling commences, all crew members begin with a fresh pair of clean nitrile gloves. To minimize contamination, the lead sampler handles the sampling collection and is considered the contaminated member, while the sampling assistant prepares the clean tubes and lids for collection to minimize contact with contaminated tubes or surfaces. While the crew nears a desirable sampling location, the lead sampler will rinse the bleach from the sampling rack in the water to prepare for loading sampling tubes, taking care to touch only the water from the sample site with the tube rack. Meanwhile, the sampling assistant will gather the 5 tubes associated with the sample and remove the caps to store them in a clean container or glove while the sample is collected. Then, the assistant will load them securely into the sampling rack. After the sampling rack is loaded, the lead sampler will reach over the edge of the vessel to fill the tubes by skimming the surface of the water. The sample should be gathered in the upper 2 inches of the surface water while avoiding large debris or sediment stirred up by the prop. All tubes should be filled completely and evenly. While the sample is being collected, the data recorder will record habitat measurements and sample data in the Field Maps app. After the sample is collected, the sampling assistant will carefully place or drop the caps one by one into the lead sampler's hand without touching. The lead sampler will screw on each cap tight and ensure caps are not cross-threaded. The capped tubes will be placed in a separate tube rack in numerical order. The lead sampler will then place the sampling rack back in the 20% bleach bath for a minimum of 10 seconds and change gloves for the next sample collection. The sampling assistant will not need to change gloves between samples since they are not touching contaminated surfaces. However, it is important to note that mistakes will be made, and gloves should be replaced any time that a contaminated surface is touched, or if it is noticed that any water is on the sampling assistant's gloves. Additionally, if a cap is dropped, it should be considered contaminated and replaced with a clean and properly labeled cap. It is important to keep spare supplies of the regularly used equipment to ensure you're prepared for any potential mistakes. While collecting samples, it is important to avoid vegetation and sediment. It may seem like these areas will contain the highest amount of eDNA, but it just ends up causing issues in the lab. Look before you dip and focus on collecting the most sediment and vegetation-free water at a site. Next, we will demonstrate the procedures for sample collection when a field blank is encountered. A field blank is collected in tandem with the next regular sample. The sampling assistant will remove the cap from the field blank and expose the contents to the atmosphere for 10 seconds. Then, we'll reseal the tube and place the tube in the sample rack. Then, the sampling assistant will remove the caps from the next sample tubes and place those tubes in the sample rack with the field blank. The lead sampler will reach over the vessel to collect the water sample while allowing the field blank to be submerged. Again, after the sample is collected, the sampling assistant will hand the caps to the lead sampler one by one, and the lead sampler will twist the caps onto the tubes. The samples are placed into the cooler as shipment racks are filled. Once a cooler is full or a sample site has been completed, add ice to cover the samples. When transferring samples, ensure the Survey 123 chain of custody forms are properly filled out with the case number, sample range, collection date, who collected, and any relevant notes. The person releasing the samples will need to sign the form, and the person receiving the samples will also need to sign it. This will need to occur anytime the samples change possession. Just remember, some aspects of sampling are not set in stone. Things like boat design, shore sampling, water bodies being sampled, etc. can alter your techniques. This is just one example of how it is done. We strive for consistency in following guidelines in the QAPP, but use whatever techniques work best for your situation, and be certain to document any deviations. This concludes the video tutorial.